The Paul Selvin Award is a special award presented by the Writers Guild of America.
Yeah, it's their time.
You know it?
I've heard of it.
It's interesting. It is given to that member whose script best embodies the spirit of the Constitution of civil rights and liberties, which and so on. So I came across this looking for a while, but your name comes first. I want to point that out. So Professor Selvin received a PhD from University of California, Berkeley, and it's funny on your website it says formally it was in physics, but really it was in biophysics. So I think that. So for example, for his thesis he was measuring rigidity of DNA and so on. So Professor Selvin has a very distinguished career with a long list of awards. Again, let me name just a few. Starting from the 1990 Office of Technology Assessment Congressional Fellow winner, AAAS Math, Media Science and Engineering Fellow, that's also 1990. Young Fluorescence Investigator Award by the Biophysical Society, that's 1999. Research Innovation Award by Research Corporation in 1999. Xerox Award for Outstanding Research in 2000. Quattro Scholar in 2000. It goes on and on. Biophysical Society, Michael and Kate Borani Award for Young Investigators and so forth. Physics only faculty scholar between 2004 and 2007. Professor Selvin is also a fellow of the American Physical Society since 2004. He was a university scholar here on campus between 06 and 07. He's also an excellent teacher. He was on the list of excellent teachers for many years, and I'll stop right here. So Paul, thank you so much for accepting our invitation.
Okay. Well, actually, I'm going to divert for a second. Turns out I'm not the most famous. Turns out there was a guy, Ben Selvin, who was I think he was the brother of my grandfather, and he's a person who has the most recorded albums of all time for anyone, because he was some conductor. And so, okay, so much for my claim to fame.
Well, on Google, you show up first. I don't know.
You got to look at Ben Selvin. I mean, he hasn't been alive for 50 years, so maybe that's it. But in any case, Ben clearly didn't know about super accuracy and super resolution, and that's what I'll tell you about today. In effect, there's two parts. There's the technique part, and then actually applying this. What we're going to focus on is basically how to beat the diffraction limit. Because the diffraction limit says you can't localize anything to much less than the diffraction limit of light. And we'll go into that in some details. And in fact, what I'm going to tell you about with a technique called Fiona is that you can actually you can tell where a single molecule is to on the order of a nanometer. So a few hundred times better than the diffraction limit of light. So this is what's called super accuracy. And then we'll also talk about super resolution, and we'll talk about the difference between accuracy and resolution. And as the last speaker mentioned, there's a couple of, by now, famous techniques called POM and STORM. And in fact, there's another technique which we will probably get time for in terms called STED. And these have become very popular nowadays. And we'll use these techniques to apply to molecular motors. That is, things inside of a cell are moving around, and all the time they have to, proteins need to move from where they're born, namely around the ribosomes, to where they're actually going to be doing its action. And in order to bring them from the ribosomes to the cell membrane or wherever, it requires other proteins that basically walk around and carry these proteins. Okay, by the way, I'm going to do something which maybe is a little against the tradition, in that I prefer, if you don't understand something, that raise your hand and interrupt me right away. Because I find, well, if I'm you, and let's say I'm going along, and I'm not really that coherent, and so you're sort of looking like that, and you're kind of like, oh. So my goal is to keep you awake over the next hour or 45 minutes. And so having a conversation is generally much better for that. Okay? So what's my name?
Ben Selvin.
Ben Selvin, that's right. I'm Paul. Why don't we start? And that is, we start with super accuracy, and this is a woman named Fiona. Do you all recognize her? Yes, okay. She was in the movie Shrek, a cartoon character. And what you may not notice is, while her name is Fiona, in fact, Fiona can stand for something, and that is, it stands for fluorescence imaging with 1 nanometer accuracy, because in fact, that's essentially what we get. We get on the order of 1 nanometer in anywhere from 1 millisecond to 500 milliseconds. And what we do is, it's really very easy. Imagine that you have a single molecule, single fluorescent molecule, and you take an image of it, and it looks like an isolated mountain because it's a single one. The width of that mountain or dye might be very large, on the order of, let's say, a mile wide. But you can actually locate where the peak is to as good accuracy as you want. As long as there's enough photons hitting your eye or hitting your detector, you can reconstruct this very well, and you can determine where the center is. Okay, so the center can be found much more accurately than the width. And in fact, instead of using a macroscopic object, this is the emission from a single molecule, single fluorescent molecule, and it has a width. That width is the diffraction limit of light, which happens to be roughly lambda over 2. Lambda is the wavelength of emission, so on the order of 500-600 nanometers. So 500 or 600 nanometers divided by 2 is 250 to 300 nanometers. But like I said, we can determine the center as well as we want to, as long as we have enough photons. And it turns out that the center you can locate to about equal to the width divided by the square root of n, where n is the number of photons. So the width is on the order of 250 nanometers. This is actually a real image, and let's say there's about 10,000 photons. So the square root of 10,000 is, come on, you gotta, okay, is 100. 250 divided by 100 is 2.5 nanometers, and it turns out it's a factor of 2 better. So you can actually get the center to 1.3 nanometer accuracy. So that's what we call nanometer accuracy. Okay? Let's actually show this, and that we're going to start with these molecular motors. We've looked at a bunch of these molecular motors, but in particular, we're going to look at this type, which is called kinesin. Turns out there are 3 types of cytoplasmic molecular motors, one called kinesin, one called dynein, and one called myosin. Doesn't really matter, but the kinesin is a dimeric motor, namely it has 2 parts held together by a coil. Each part can bind ATP and can cause it, upon binding ATP and then hydrolyzing it, taking some of the gas and basically going with it. Just like your car takes some gas and it turns the wheel. What happens is it takes some gas, some ATP, and in this case actually causes it to walk. When it causes it to walk, the cargo, which is up here, which we're using just a quantum dot, which is a very fluorescent object, or we can use a regular fluorescent object, that the guy will move. In this case, for kinesin, it moves 8.3 nanometers, so very, very small. So can we see this? And in fact, there we've labeled a single kinesin with a quantum dot, and it's walking on what is called a microtubule, and we can see them. Can we actually get out the quantitative information? Yes. What we do is we plot as a function of time the position, and here it's not moving, and all of a sudden an ATP comes, it takes it, it chews it up, and it causes it to walk. And then another one comes, and then another one comes, and then another one comes. And in fact, what we can do is we can look at the step size as a function of just counting, and you can see it fits a Gaussian, very nice, to 8.4 nanometers plus or minus 0.7 nanometers. So what we have is basically the fluorescent object, which in this case is on its head, is walking 8.4 nanometers per step. Okay? So that says with an uncertainty of 1 nanometer. Okay? So that's my first claim, that we can get super accuracy. So is there any questions? Yes.
So how rigid is this kinesin? Is it like dragging something by a string, or is it more broad?
Well, in this case, in some senses, it doesn't matter. In the sense of what happens is it takes like this 8.4 nanometer. Presumably, it's undergoing some constrained diffusion, but all of that is happening much faster than the time scale. And in fact, the time scale for diffusion is on the order of microseconds, or faster, where in this case, we're taking it with a millisecond or tens or hundreds of milliseconds. So all of that is basically averaged out. So in fact, whether this, when we were first starting this, we thought, oh, maybe the diffusion is going to kill us. And it turns out that that's not true. And it's basically because the diffusion is much faster than the time scale for recording it. It's a very good question, though. Yes?
I was kind of curious about the time frame. So how, when do you take each image?
Yeah.
What you basically do is you turn on the light, and the fluorophore is there and is emitting, and we take enough time that we can get a reasonable number of photons. In this case, if you're getting, let's say, about 5,000 photons per second, as we did in the last one we showed, we can get about a nanometer accuracy. So that, in fact, that's how long we take it. Initially, that meant that it was about every half a second, so fairly slow. The way we did that is we limited the amount of ATP added. So it took a step, on average it waited, took another step, and so forth. It turns out with quantum dots, one of the real advantages is they're so bright that, in fact, we went down to about 2 milliseconds.
And what size is there, like, do you know?
Right. At first, it doesn't matter, but if you use the commercially available quantum dots, they're about 20 nanometers. They're really actually huge. In fact, it's huge compared to the distance. It turns out, which I'll talk to you in a little bit, we've developed new quantum dots that are about 7 or 8 nanometers, so about half the size in terms of diameter. Yes?
You something that was strong, like some sort of background in your image. So where does it come from?
This is for presentation purposes. You want to illuminate the track over here with light, so you actually do it in bright field. So that's where the background is coming from. Though in reality, the fluorescence that you see is basically on a dark background. It turns out, actually, the issue of background in general is very real, in particular looking at cells. And in general, you want to decrease that as much as possible. But to a certain extent, the fact that you have background, it basically hurts your accuracy. Okay, that's fine. Last one.
If you want to image faster, you hit it with more light, but is there some saturate your quantum dot? What is that limit?
Exactly. If you want to go faster, you hit it harder, you get more photons out. At some point, actually, what happens is you saturate your quantum dot. That is, you go and excite it, it goes up, and then when it comes down, it emits a photon, and you go like that. At some point, you knock it up, and then you actually knock it up again before it can come down. So in this case, the limit was about 2 milliseconds per point. Also, it turns out what happens is the kinesin is relatively immune to this high light, but actually not completely. So actually, what happens is you get you try to hit it with getting a lot of light out, the kinesin starts to go bad. With these biological systems, that's a pretty common phenomenon, that you want to hit it, but without actually causing inadvertent damage. Okay, not only can we do that, but so in there, what happened is we were having a quantum dot on the cargo domain. You can also put another one on the foot. And in fact, what happens, if you think about it, it moves 8.3 nanometers ahead. But now, if you put a fluorophore or another quantum dot on the foot, how far is that going to move? How far? Twice, exactly. So it'll move 16.6 nanometers, and then 0, and then 16.6, and then 0. And that's exactly what we get. Okay? No. Now, one further interesting application of getting super accuracy. It turns out that in your body, for example, there are nerve cells. And all the time, over here is what's called an axon. And in the axon are these microtubules, the roadways of which it's walking. And notice this cargo here is not just walking in one direction, but it's walking in both directions. And it turns out if you have a roadway that you have kinesin, which is walking north, let's say. Now, what if you wanted to walk south? It turns out there's another molecular motor called dynein, which walks the other way. And so the question, and apparently it's very, jiggly. It goes this, and then there, and then there. It turns out that if you have a cargo in your body, it typically has kinesin and it has dynein. So how does it decide which way to go? I mean, they both use ATP. And there's two types of models for this. One is a tug of war, where what is happening is kinesin is pulling as hard as it can, and dynein is pulling as hard as it can, and whichever one is stronger wins out. Okay, that's called a tug of war. That makes sense, but it's an awfully inefficient way of utilizing ATP because, you have ATP being used this way and ATP being used that way, and you're not really getting very far. So, in fact, a coordinated model was invoked, and that is there's some external protein or hormone or something that comes on and attaches to the kinesin, and the kinesin says, let's go, and dynein takes over, and then at some point the protein or hormone comes and binds to dynein, and it lets go, and kinesin goes. That's called a coordination motion model. The problem with that is basically people spent about 10 years looking for one of these coordination models of protein or hormone, and they could never find it. Okay, and it turns out that there's another method, which I think I don't have time for, but it's called a synergistic model. That is, maybe it's like the tug of war. Each side are just pulling on it, but in fact, what happens when kinesin goes along, maybe the dynein comes off, maybe it doesn't. Maybe it actually follows behind. And then when dynein comes on, that kinesin falls off, for example. But we're not going to really, for now, differentiate this. But what I'm going to talk about is the tug of war versus the coordinated motion model. And in particular, notice that the coordination model has an external protein or molecule which is doing the coordination. So what we're going to do is we're going to take a fluorescent object, fluorescent so we can see it. We're going to attach some purified kinesin and some purified dynein, no external regulatory, and we're just going to look at it. If, in fact, we find that it undergoes this up and back model. So then a coordination model, at least at first, doesn't appear to be correct. Okay? And in fact, well, that's exactly what we do. And in fact, what happens is it goes dynein and kinesin, then dynein and then off, dynein, kinesin, dynein, blah, blah, blah. So in other words, that in fact it is not a coordinated motion going on. It's some sort of tug of war. Okay. Is there any questions at this point? Because we're going to, I believe, at this point, go to super resolution, which we'll explain in a minute just what that is. Okay. Okay. We've talked about getting very good accuracy and locating things. Now we want to get very good resolution. That is much better than lambda over 2 or 250 nanometers. Resolution is when you don't have a single molecule, but you in fact have two molecules or many molecules, and you want to tell the distance between the molecules. Typically, what happens is you have each forming a little mountain, so to speak, and as long as that the distance between the mountains or the dyes are sufficiently far apart, that is, this would be about 250 nanometers and this would be about 250 nanometers. As long as they're far apart, you say they're well resolved. As these guys become closer together, they don't become so resolved so well, and this is called the Rayleigh criteria. This is about lambda over 2. And if you go even farther, they're not resolved at all. Okay? So, how do we do this? Actually, it turns out it's very easy, because here we have it's unresolved, and you're shining light at it, and basically two of them are fluorescing. But at some point, one of them dies. In this case, now you have one molecule. You can, with Fiona, you can determine where the accuracy to, let's say, a nanometer is. You have this to a nanometer. Then you go back and you look at the so-called unresolved spot, and you take the unresolved minus the resolved, where this guy is gone, and the difference will be equal to this guy. In fact, and then you can fit that and get it something like to a nanometer, and basically you've separated the distance to on the order of a nanometer. Or, in fact, another possibility is maybe the thing doesn't permanently photobleach, it temporarily photobleaches. So one guy's here, then one guy's gone, and then later on this guy disappears and this guy reappears, and in which case you can do Fiona on either of them. Okay, so what we have as a test is you have some DNA with a dye on either side, and at first you shine light on it and both of them are alive, and so you have 2 units of fluorescence. Then at some point one of them dies, and you should get half half the units of fluorescence, and then at some point it completely dies. And then you should be able to reconstruct. Okay, let's see if we can do it. Okay? So there's 2 units, and then boom! There's 1 unit, and boom, 0. And you should be able to see the difference. In fact, if there's 133 nanometers, we can see that. In fact, if we make a smaller piece of DNA, we can see 72 nanometers. And then if we go really small, we can see less than 10 nanometers, the difference between 2 dyes. Furthermore, what we can actually do is not only have 2 dyes, but actually we could have 3 dyes. And so at first you're hitting it, you'll get 3 units of fluorescence, and then you get 2, and then you get 1, and then you get 0, and then you build back up. In fact, you can keep going. Why not do 4 or 5? And that is in fact exactly what we've done, and we can get to something like 10 of them, something like that. Yes?
So this switching on and off of adjacent workloads, and when you control it somehow, it's not random. The person is thinking, how does that happen?
Yeah, with this technique, it is in fact random in that that is, if you have 3 molecules, you know, in general at first all 3 are fluorescing. One of them dies. It could be this one, could be this one, this one. You don't know until then 2 have died and then 1 have died, and you can reconstruct them. There is no control over that. Whether you do this type of technique, where this is shrimp, super high resolution imaging with photobleaching, or where you have actually 1,000 of them, and we'll discuss how to specifically turn on and off the molecules, though which one you're turning on and off, you don't know. Exactly the same. Exactly the same. And it purely is a matter of stochastic. That's basically saying, okay, you have 2 molecules, and which one goes, you have no idea. But one of them doesn't go. But both of them coming and both dark, and that one happens to excite a molecule and goes, and the other one sometimes goes, and again, it's random. And that is, you don't turn on your laser light to the point where, you know, the thing, because all of them would go within like a second or something. You have a reasonably weak light such that, you know, you have this guy going and this guy going, and then randomly.
But both of them are getting same contact. And both are similar. If you say that one is other dye and the other is the other dye, and one is very sensitive to photobleaching, I think the first is getting off and the other is.
Right. But what you actually have, if you have either even really good dyes like rhodamine or Psi3, that sort of thing, there is always a distribution in terms of what's called the photobleaching lifetime. Some will last after they are emitting 1 million photons, some after 900,000, some after 800,000. It's random. And the chances are that any 2 will actually die at precisely the same moment. In general, it's fairly small. It turns out it's not 0, And in that case, actually, what you end up seeing is some fluorescence, and then boom, it goes to 0. And then you don't count those. But the other ones, which somehow do, like that one, go 2, and then 1, and then 0, those you do count. Yes?
So do you think you can build like similar fluorophores but with different quantities so that you can control it?
Well, you get enough sampling. Yeah, though you can't control it. Even if you built 2 fluorophores were the same, but one varied, you know, which on average would die after 100,000, and one would be after 1,000,000. So in general, the 1,000,000 would last longer than the 100,000. But remember, it's in general because the first one will be an average of 100,000. I mean, if it's purely binomial distribution, it'll be plus or minus the square root of 1,000. And the other one will be 1,000,000 plus or minus the square root of 1,000,000, and so there'll be some little overlap. So it turns out it's basically not worth it. Okay? Okay, so in this case, we're going to be looking at nerves. And we're going to be looking at super resolution nerves, and that in your brain, but everybody seems to be awake, but listen to me for the next minute. This is the most unbelievable thing. In your brain, you have about 180 to 100 million wait, I don't want to mess this up. Just getting confused between a million and billion. Yeah. Billion. Okay, there we go. Okay, thank you. So you have between 80 and 100 billion nerve cells in your brain. That's quite a number. Each nerve cell tends to connect to about 1,000 other nerve cells. Your brain is extremely intricate. So that means, and the connection between them is via synapse. It turns out one nerve cell connects to another nerve cell and actually doesn't connect, it actually has a small space, about 30 nanometers across, but nevertheless. So that means you have 80 to 100 trillion synapses in your brain. I mean, it basically puts computers to complete shame. And now, if you have a disease like Alzheimer's, what happens is these nerve synapses go bad. Or in my case, whatever, 13 years ago, I was hit by a car, and I had a stroke, and so there was a brain bleed, and basically it made it so that certain synapses were destroyed, and so I walk with a little bit of a limp. Okay, so it's clearly of great clinical significance what these synapses are. What we're interested in doing is we're interested in knowing where some of the proteins in the presynapse, that is part of the synapse of nerve 1, and some of the protein in what's called the postsynapse in nerve 2. And here that shows the synapse is just 30 nanometers across. So one thing is, and the synapses are sometimes oriented like this, nerve 2 and nerve 1, sometimes like this, sometimes like this. So in fact, we would like to visualize this with not 2D resolution, but actually in 3D resolution, super resolution. This here is generally about a diffraction limited width apart. So what we want to do is see individual proteins moving around. In order to do that, we need small and photostable probes, because they have to get in here to be able to label it. So they basically have to be smaller than 30 nanometers, where in fact, there's a question of how big are the fluorophores, and it turns out that big quantum dots, that is commercial quantum dots, in general are too big to fit in general. Okay, and furthermore, what we want to do is we want to look at live cells, because one of the things about learning is these synapses are strengthened or forgetting, they're weakened. And so it turns out that the way that a synapse is strengthened or weakened is basically by these AMPA receptors. In terms of, they'll get a lot of the AMPA receptors come into the synapse, and that's you learning. And the AMPA receptors then eventually fading away, and that's you forgetting. I mean, it's kind of amazing that you can take this incredibly complicated system, 100 billion neurons, and yet you can then say this really complicated process of learning and forgetting, and it comes down to whether these AMPA receptors are inside the synapse and communicating, which I find incredibly fascinating. Okay, and what we're going to do is determining some proteins in here in the presynapse associated with nerve 1, and in the postsynapse associated with nerve 2. Okay, the way we're going to do this, by the way, the reason you need resolution is, for example, if you label these proteins in the postsynapse over here, there are many, many of them. And so you have to individually localize all of them. Okay, and what we're going to do is use PALM or STORM, and this is by far the most common popular method. And let's say you have something like this, but in general, this is smaller, on the order of the diffraction limit of light. Diffraction limit of, that's 250 nanometers or so. And if you shine light at it, each of those points emits and creates an image which is about 250 nanometers, and so you get something that looks like that, which is pretty lousy. Okay? However, let's assume that all the fluorophores that you have on there are dark. And what you do is you come on with a little bit of UV light, and you randomly will activate certain ones here and here, just randomly. Because in general, there happens to be enough light here and not next to it. Again, it happens, it's random. Okay? So you've activated it, and then what you do is you turn on some excitation light, which in general will be in the visible light. Again, you then have this guy emitting and this guy emitting, and as long as they're far apart from each other, you can actually determine where the center is of this is using the Fiona, using the very good accuracy to about a nanometer, and same thing in this, and some things like that. So in fact, what you've done is you've isolated this one and this one and this one, and you've gotten them to a nanometer or so accuracy. And then essentially you just repeat the process, and the next time you have this one and this one and this one or whatever, and you can actually spell out what the original image, even though these guys may be only a few nanometers apart. And that's the fundamental idea of super resolution microscopy. And it's important that you know this. So is there any questions? Yes, thank you. Yeah.
Does the near UV light activate some probes?
That's a good question. Next question. It turns out that basically it was initially done through luck. And it took several years for them to actually figure out the photophysics of it. In the case of GFP, there's a special mutation associated with GFP that it makes it dark, and it enables you to activate it. Yeah. Next question. Yeah, okay, you got me one.
So it's different for different fluorophores, and do you need to have a specific color fluorophore? Like, does it only work for green because you blue shifted it by this amount, or can you do it with red?
You can do it for red ones as well. In general, you use like 4 or 5 nanometer to activate it, and you can use 4 or 5 nanometer to activate a mutant of GFP, or you can use 4 or 5 to activate like a CY5 or that sort of thing. In general, what has happened over the years is, in fact, they've managed to have more and more dyes that actually work with this. Yes?
That we have to shine the light, excitation, and like 4 come on, and then the next frame, another number of them are shown. In order to get a full image, we will need enough time to have them all turn on. So does that make this technique not very suitable for getting a complete picture of something that's happening fast?
Yes. That's exactly right. Though again, over the years, people have pushed this such that initially it took like 24 hours in the initial science paper for them to get an image. They've now pushed it such that it's about 20 minutes, which is still long. And in fact, they've pushed it now that we typically, when we're in a hurry, we can do it in about a minute or 2. But faster than that, you're in big trouble. And in fact, I think we probably won't get to it, but STED is another super resolution technique which doesn't have as good accuracy or resolution as PALM or STORM, but STED is inherently really fast. In fact, STED, you can get about 40 nanometer resolution at 30 frames per second, which is impressive.
So, generally speaking, for live cell imaging, you want to use STED. For pixels, you want to use PALM.
Well, yes and no. In fact, we've done live cell imaging with PALM because that we're in somewhat of a rush, but not that much of a rush, and that it turns out that 2 minutes is enough time to get, in fact, the postsynaptic density, because basically the proteins are there and they're not changing in the course of a couple of minutes. So it's fine. And then it turns out we do Fiona in terms of looking at the AMPA receptors, and that we want to get some number of millisecond time resolution.
In the series of activation, first is brain. So the one effect function is not overlapping, so you are able to resolve.
That's right.
But in the next series of brain, when there are two quantum function already
well, you have to remember, you read it out with visible light, and you get this. But it's important is the molecules are no longer photobleaching. In fact, you typically wait with visible light, you wait till the original ones are all photobleached. In fact, that's another thing that you have to do. That's a very good point.
Unless you have a switch on.
Yes, there is nowadays a switch off light to make sure that these are not still illuminating, which it is somewhat useful. Okay, and now, so this is, we're going to be able to get 10 or 20 nanometer resolution. Now, so far, what I've only told you about is how to get 2D resolution. That maybe the molecules are extended in 3D. It turns out that that is actually very easy to do. What you do is you put in a cylindrical lens, and what happens is if the image is right in focus, the X and the Y look the same. However, if the image is up above it, then the X happens to be much greater than the Y, and if it's below it, down here, then the X is very small and the Y is very big. So basically, you do the same thing, but with a cylindrical lens in it, and by looking at the ratio of X and Y, you can determine what Z is. Okay? Okay, and so in fact, we've done this. In this case, we took an image of Homer 1. It turns out that's a postsynaptic density protein in the nerve 2, and in nerve 1 was another protein called synapsin. And you see here in blue and in red, and in particular for example, in A or B or C, right here is the synapse. And this is in 3 dimension. Okay? And this is in live cells. So the thing to really do is to treat the cells to essentially make them learn or make them forget, and see if whether these support structures, these proteins here, these proteins here, increase or decrease. Or, in fact, again, what we're really interested in looking at is the AMPA receptors. And in fact, to look at the AMPA receptors, we're going to attach a big quantum dot, that is, quantum dots that are commercially available, or we're going to label them with small quantum dots, and we're going to see what the difference is. And it turns out the big quantum dots will have a difficult time getting in. The way we make these small quantum dots is you have a quantum dot core, which is generally something that is super fluorescent, although it's fluorescent only in like organic solvents. If there's water around, water will contact here and completely quench it. So what you have to do is you have to protect it. In this case, you protect it with like an 11 carbon atom. You connect it via a metallo-sulfur bond. And then, in fact, because you're working in water solution, you have to make it soluble in water. So we add what is called PEG, polyethylene glycol, at the end. And in fact, you see this one is about 6.3 nanometers in size. Typically, what happens is you have what is made by companies is you have about a 5 nanometer core, and then you have some massive shell. The massive means it's about 20 nanometers in size. Okay. And furthermore, we've made this in different colors. You change the core shell, and we've gone at 527, 560, 615, 650. And in fact, they're easy to see with the single molecule level. Okay? Okay, so now we're going to do tracking. Oh, here's one dendrite, and here's another dendrite, dendrite being a part of the nerve cell. What we've done is we've done PALM to locate where the synapses are. This happens to be Homer 1. And then we add a quantum dot, which is excited at a further red wavelength. And we'll see that we get. And this is on live cells with 3D resolution. There you see the, and in particular, we see the AMPA receptor, the Homer 1, and then look, here is a single AMPA receptor labeled with a quantum dot. Notice actually it's sort of going all over the place. This is something which is in fact completely new information. The frame rate, I think it's either 10 hertz or maybe 20 hertz. Okay, so this is with big quantum dots. That sometimes apparently the big quantum dots are small enough to get in, or the synapse is big enough that the big quantum dots can get in. But in fact, mostly they're constrained to being extrasynaptic. As you can see, this is one molecule. It's walking all over the place, and mostly it is not getting into the synapse. And it turns out that only about 15% of the time does it get in the synapse. So what I'm saying is the big quantum dots are mostly diffusing outside of the synapse. This is actually important because people have chosen big quantum dots because they're incredibly bright and you can actually see them. With regular fluorescent molecules, which are, you know, like 2 nanometers in size, they were good because you can see actually in the synapse, but they're not photostable enough. Basically, they die after like a second or two. Okay? So this says that big quantum dots have somewhat of a problem. Now we want to turn to small quantum dots. Again, they're about 8 nanometers or 7 nanometers. We label it an image, the postsynaptic density, Homer 1, in blue. And we also label the AMPA receptor in red. And again, we can get 3-dimensional resolution.
How long does it take to capture this entire thing?
Okay, I'll be with you in a second. Okay, so this is the Homer and this is the AMPA. And notice, again, it's diffusing around. And actually, it's only diffusing actually in a part of the receptor. It turns out to get this in terms of doing the palm on the Homer, it takes about 20 minutes if we're not in a big rush. That is, if we are in a big rush, then we can do it down to 2 minutes at somewhat worse resolution. And that's the Homer. Then the AMPA receptor, we typically, again, 10 or 20 times per second. Okay. And in fact, we can get, as we said, the 3-dimensional orientation of this. Okay, so the first thing you notice is that the AMPA receptors, they're in what are called these nanodomains. They seem to be in only a part of the synapse, so that's new. But in the case of small quantum dots, about 85% of the small quantum dots can get into the synapse. And what's more is this is a fairly amazing slide. Here you see a dendrite, and here again, this is the postsynaptic density, and here's the AMPA receptor going in a nanodomain. But more to the point is you're not seeing a tremendous amount of diffusion. That is, most of the AMPA receptors are not coming from outside of the synapse and then going directly in. In fact, most of the AMPA receptors has to be coming internal to the cell membrane and then coming up. and again, these are some examples of various nanodomains.
Are there any synapses that you reject because you can't extract them in your field of view?
Yes, in the sense of, for example, here, there's apparently an AMPA receptor going around, and there does not appear to be a homer because there's no blue there. Why that is because either the homer didn't happen to get labeled, or because the AMPA receptor happens to bind there, and there's some other reason that it's binding, we actually don't know. So in fact, for now, we're not very interested in like that. But this is actually an unreconstructed image of all of the AMPA receptors there. So they're clearly not here and not here and not here. Now, whether we can determine with very good accuracy on any one particular one, we actually haven't done. So in fact, we cannot say what percentage of the synapses are in these nanodomains. It is definitely not the majority, but it's clear there's a significant number. But what that number is, we actually don't know yet. Yes?
We currently have quantum dots that would bind, fluoresce, and turn off. In other words, can we tell whether these points that we're recording are different receptors or the same receptors diffusing through the membrane?
Yeah. It turns out, I don't have it here, but occasionally, it's very rare, but you will have a quantum dot on a receptor, and then it will apparently get endocytosed, move along, and go to another one, and then get endocytosed and move to another one. We've actually have seen that, and we don't know the significance of that yet, but it does imply that there is a certain amount of intersynaptic communication, which actually, you talk to some bigwigs in the field and they say, oh yeah, that's really well known. But then you actually talk to others and, oh no, and that can't possibly be. So, okay? Mm-hmm.
Curious how you visualize the boundary of
in general, actually, we don't. What we do is we see that this is what's called a dendrite, which is a continuous part of the nerve cell. In fact, in the future, we're going to label this with some sort of regular dye and see that. But that would be better.
The membrane of the synapse itself, that blue border?
No.
Is that real, or is that the probability?
That this is so-called not real, and that this is where the homer is. This is on a nerve cell, and so there is a cell membrane going over here, and at each of these there actually is another nerve cell coming in, although those are not labeled, so we generally don't see those. Okay, and at this point, I think we won't cover STED, but as I mentioned, it is another technique to get super resolution microscopy. Essentially okay, I'll go for a minute on this. Usually you get like on the order of lambda over 2 or 200 nanometers. If you hit it at a further red over here, you can suppress the fluorescence from over here, and so you get this minus this, and you get effectively this. And so what you do is you get a point spread function on the order of 20 or 40 nanometers, and so you actually can get that kind of resolution, and it's not on single molecules. That's enough. And here is getting 35 nanometers, normally with 190 nanometers. Okay, so this is basically a conclusion of what I've said, and the end.
Okay, any other questions?
You've been in the field for too long. What's your...
You or I? Oh, I've been in the field too long? Okay, I'll retire.
What's your estimation of when we will be able to do this 3D in vivo?
Well, it's a question of what you mean by in vivo. This is in vivo in the sense of it's on live cells. No, but, right. Oh, okay. When can we actually start looking at animals and that sort of thing? That will keep me and other people busy for a long time. Yeah. Whether it will happen before I retire, well, I guess it's your guess as to how old I am, and I don't know.
Just a comment. I mean, in the STED microscopy, for example, right now, people have developed really some techniques that you can look at multiple planes at the same time, and it's fairly fast. There's no reason why you shouldn't be able to do that in vivo, like, like if you open a house install and install the microscope, similar to the experiment we saw like the other day. That's technically in vivo and it's fast enough to study. Of course, it's happening, it's going to be deep work within the realm of the non-optical microscope assessments that we do the English letters.
Right.
Except it's cooking.
It's what?
Except it's cooking tissue.
Yeah. Okay. That's the basic problem with STED, is that it requires, in general, very high light intensities. So there are very few fluorophores which are okay with that, and that you tend to cook the tissues. Now there has been a lot of work since STED initially came onto the scene as to how you can decrease that amount of light, and well, and that's why STED is still in the ball game as to.
I just want to add to that. Basically, of course, the original concept of the STED, if you just want to basically bring them down to the ground state by dumping lots of intensity into the tissue. But the original concept of the STED is basically using nonlinear optics. And if you, and there are again papers, recent papers, that if you do this on-off switching to other mechanisms, if you use smarter basically fluorophores or photoactive or photoswitchable probes, you can use probably 1 million times lower intensities, and it still gets over resolution, so that doesn't necessarily.
Right, you're absolutely correct. What type of resolution you get is fairly unclear, in that typically people will get actually something like 100 nanometers, or if they're doing really well, they're getting like 50 nanometers, which is still good. You know, that's up to a factor of 5, so you still call it so-called super resolution. It does not get down to 10 or 20 nanometers where the Palm or Storm is getting. And what I find is there's the resolution gotten by basically Stefan Hell, which is generally a factor of 2 or 3 better than anyone else. And that's because he's really, really good.